Hello everyone, my name is Kay Wilson. On behalf of Asterand Bioscience, I'd like to welcome you all to today's webinar, the Human Islet Organ Dot System. First and foremost, a big thank you to everyone who has registered for this webinar, enabling us to make a substantial donation to Diabetes UK and the American Diabetes Association. I'd like to mention a few points before we begin. Please note that all attendees are muted. To ask questions or share comments, please use the questions and answer panel on the right-hand side of your screen to chat with us. Please also use this chat feature to alert us to any audio-visual issues that may arise during the webinar. The webinar will run for approximately 25 to 30 minutes, which will be followed by a question and answer session. You can send your questions at any time during the presentation by typing into the chat box on your screen. Please send your questions in as they occur to you and we will collect them. Our speaker will try to get to as many questions as possible during the question and answer time after the presentation. This webinar will be recorded and hosted for future reference on the Asterand Bioscience website. I would now like to introduce our speaker, Dr. Graham Place, who is the Director of Scientific Operations for Asterand Bioscience, based in Royston in the UK. Over to you, Graham.
Thank you, Kay. Hello, and thank you for joining our webinar on the Human Islet Organ Dot System. Asterand Bioscience is especially pleased to be able to present this webinar in support of World Diabetes Month. My first slide just provides a brief background to Asterand Bioscience. Asterand is a market-leading provider of human tissue and tissue-based research services. Formed in 1996, the company has a strong background in research services and in the procurement and supply of human tissues and derivatives to our clients. Our tissue products business is based in Detroit and specializes in the procurement and supply of human tissues in various formats, such as formalin fixed, paraffin embedded blocks, or snap frozen, with a wide range of samples available from diseased or non-diseased tissues. Asterand's research services business is based in Royston and offers expertise in several areas. For example, target validation using cutting-edge immunohistochemistry and in situ hybridization techniques, complemented by computer-aided image analysis, compound profiling using human primary cells, and isolation and supply of human primary cells from normal and diseased tissues. We have a global tissue procurement network and currently have more than 100 active sites. Our work is underpinned by strong quality management and regulatory systems. And finally, we work with most pharmaceutical and biotech companies, along with many diagnostics companies, academics, and US government agencies. As I mentioned at the start, Asterand is pleased to present this webinar in support of World Diabetes Month. Diabetes presents a huge global healthcare challenge, as about 400 million people worldwide are diagnosed with the disease, and it is estimated that 5 million people die each year of diabetes-associated complications. Diabetes is a group of metabolic disorders characterized by poor glycemic control due to defects in insulin secretion, insulin action, or both. Most importantly, chronic hyperglycemia is associated with long-term damage, dysfunction, and failure of organs and tissues such as the eyes, kidneys, and nerves. There are two major forms of diabetes, type 1 and type 2. Type 1 is an autoimmune disease that leads to destruction of the insulin-secreting pancreatic beta cells. Only about 5% of patients suffer with this form of diabetes. By far the most common form of diabetes is type 2, which is a heterogeneous metabolic disorder characterized by reduced insulin secretion and reduced sensitivity of peripheral tissues to the hormone. Whilst the effects of diabetes were known to the Egyptians and ancient Greeks, the discovery at the turn of the 20th century of the role of insulin in maintaining good glycemic control and what happens when blood glucose is not properly controlled led to a better understanding of how to treat the disease. Initial treatments were based on diet and exercise, and lifestyle changes remain an important element of control for people with type 2 diabetes. However, a careful diet and exercise alone is not sufficient to maintain good glucose control for many, perhaps most people with type 2 diabetes, and therefore numerous drug treatments have been developed. These include peripheral tissue sensitizers such as metformin and glitazones, agents that promote insulin secretion such as sulfonylureas and incretins, and drugs that inhibit the action of the kidney glucose reuptake channel protein, SGLT2. Of course, a key drug for diabetes is insulin itself, critical for people with type 1 diabetes, but may also be used for those with type 2, which harks back to the pioneering work of Frederick Banting and Charles Best in the 1920s. Despite the array of drugs currently available to treat diabetes, there remains an urgent need to identify new and more effective treatments to combat the looming health challenge presented by the disease. The search for new insulin secretagogues continues, and this has driven our research to develop and validate a reliable and robust in vitro test system for such compounds, the human isletOrganDOT system. First, I'd like to remind you of the intracellular mechanisms of insulin secretion. This cartoon, taken from Sino and Shibasaki, shows the key events involved in insulin secretion. External glucose is taken up by glucose transporters and is then metabolized, ultimately leading to inhibition of ATP-dependent potassium channels. The subsequent depolarization of the cell opens voltage-dependent calcium channels, leading to an influx of calcium and stimulation of the secretion of insulin granules. Activation of the GLP-1 receptor, a GS-coupled GPCR or GQ-coupled GPCR such as GPR40 are alternative mechanisms for promoting insulin secretion that work in concert with the effects of elevated glucose. A number of models have been used for the investigation of insulin secretion, and each one has its advantages and disadvantages. Cell lines such as Min6 are popular because they provide a convenient and readily available system for studying insulin secretion, but they have only partial beta cell function and tend to lose differentiated properties in long-term culture. Great progress has recently been made with stem cell-derived beta cells, but fully differentiated cells that have the same secretory capacity as native beta cells are not yet readily available. Nevertheless, they remain the focus of considerable research efforts, particularly generating cells for transplant. Fresh rat or mouse islets retain good beta cell function, but the cellular architecture of rodent islets differs markedly from human islets and thus raises questions over the relevance of this model for testing compounds to be developed as drugs for humans. Human islets represent the gold standard for in vitro studies, but limited supply, high cost, and rapid loss of function post-isolation are major disadvantages. Despite the non-trivial disadvantages of using human islets, these remain the test system of choice for evaluation of lead compounds as insulin secretagogues. Insulin secretagogues are usually tested in glucose-stimulated insulin secretion or GSIS assays. A typical GSIS assay involves initial equilibration of fresh isolated islets in a medium or buffer containing a low concentration of glucose, followed by switching the islets to a medium containing a higher concentration of glucose in the presence or absence of the test compound. A known secretagogue, such as the GLP-1 receptor agonist Exendin-4, which is a drug marketed under the brand name Byetta, is often included as a positive control. Samples of the solutions from the low glucose and high glucose incubations are then assayed for insulin. In principle, this is a straightforward assay, but apart from the high cost of isolated human islets, there are several problems associated with using them. Perhaps the main one is rapid loss of islet function within a few days post-isolation, which is particularly problematic for users who do not isolate their own islets. Islets are naturally of different sizes, and this influences the number of beta cells and hence the amount of insulin secreted in response to raised glucose or other secretagogues. To overcome size heterogeneity, Some users pick islets of similar sizes before performing GCIS assays, but this is very time-consuming. Using intact islets also has the drawback of being low throughput, so testing several compounds with the same batch of islets can be difficult. To overcome the shortcomings of using intact human islets for GCIS assays, we developed the isletOrganDOT system as a robust, reliable, and relevant in vitro model for the evaluation of insulin secretagogues. The model is based on the OrganDOT methodology for creation of 3D microtissues developed by Capstan Neurotechnologies. The proprietary rights to this technology are now assigned to Asterand. This slide illustrates how OrganDOT cultures are produced. Freshly isolated islets are dispersed with protease and trituration through a syringe needle, which breaks up the islets into individual cells. The cells are washed by centrifugation and then seeded at high density in a microvolume drop onto porous membrane inserts. Medium is only present on the basolateral side of the membrane, creating an air-liquid interface. The interface, combined with capillary compaction, drives the cells to reaggregate to islet-like structures that retain functionality. Generally, we prepare isletOrganDOT cultures on 24 insert plates, which allows for convenient GCIS assays. This slide shows how the dispersed islet cells reaggregate over time after seeding onto membranes. On day 0, you can see a lawn of isolated cells, but by day 1, the cells have already started to aggregate, as shown here. And this aggregation continues with time in culture, so that we start to see clear aggregates of islet cells, and by day 12, we have clear 3D type structures. Typically, we generally use isletOrganDOT cultures 5 days after seeding the dispersed cells. To best mimic natural islets, it's important that the OrganDOT cultures retained the normal complement of islet cells, and these confocal immunofluorescence images of stained alpha, beta, and delta cells show that these 3 key cell types are present in the aggregates. The top panels show native pancreatic islets immunostained for various islet cell marker proteins. Insulin is visualized as red, and the other marker proteins as green fluorescence. Overlapping distribution appears as yellow fluorescence. The images clearly show co-expression of insulin, proinsulin, and C-peptide in the same cells, i.e. beta cells. By contrast, expression of glucagon, secreted by alpha cells, and somatostatin, secreted by delta cells, are clearly different to that of insulin. The same staining patterns are present in the isletOrganDOT cultures, shown in the lower panels, indicating that the alpha, beta, and delta cell types phenotypes are present in these cultures, and that the OrganDOT technique does not favor survival or distribution of a particular cell type. GCIS assays are performed by sequentially transferring isletOrganDOT culture inserts into buffers containing low or high concentrations of glucose. As for the culture conditions used, only the basolateral surfaces of the cultures are bathed with buffer. Cultures are initially equilibrated with buffer containing low glucose, typically 1.67 millimolar. After 3 changes of equilibration buffer, the cultures are then shifted to a final low glucose incubation buffer for 30 minutes before a final shift to the test condition, which is buffer containing the same or a higher concentration of glucose, e.g. 6.7 or 16.7 millimolar, in the presence or absence of test compound. These basal and stimulated solutions are retained and assayed for insulin by ELISA. GCIS data are represented as absolute insulin secretion or as net insulin secretion, which is calculated by subtracting basal secretion from that measured in the test solution. Thus, each culture acts as its own control. This graph shows the results of a typical GCIS assay. There is always a low level of insulin secretion, even in the presence of low concentration of glucose, but you can clearly see when the glucose concentration is increased, in this instance 6.7 millimolar or 16.7 millimolar, there is a large increase in the amount of insulin being secreted by the cells, which is the expected response of functional beta cells. We have now performed successful GCIS assays with isletOrganDOT cultures prepared from islets from over 60 donors, and this slide shows data from 10 of the donors. As you can see, we observe consistent and robust responses to glucose. It's our experience that there is a wide donor-to-donor variation in the amount of insulin secreted when isletOrganDOTs are stimulated with glucose. For example, in the presence of 16.7 millimolar glucose, insulin secretion by islets from donor 1 is considerably lower than that from donor 3. We believe that this is the result of natural donor-to-donor variation in the responsiveness of normal beta cells to raised glucose, and not that there are fewer beta cells present in the low responders. To illustrate this point, we carried out GCIS assays with cells from 2 donors, where the OrganDOT cultures were seeded at different cell densities, ranging from 15,000 to 45,000 cells. The absolute amounts of insulin secreted by the cells from these 2 donors were quite different when the cells were challenged with 6.7 or 16.7 millimolar glucose, with isletOrganDOT cultures of cells from donor 46 secreting up to 3.5 times as much insulin as those from donor 45. At the end of the GCIS assay, the cells were lysed to measure the remaining intracellular insulin. Secreted insulin was then calculated as the percentage of the total available, the sum of secreted and intracellular insulin. The absolute amount of insulin secreted by the beta cells in both cultures was less than 2% of the total available, even in the presence of 16.7 millimolar glucose, which is consistent with published findings. Furthermore, the amount of insulin secreted as a percentage of the total was independent of the cell density, or, to put it another way, the number of beta cells present in the cultures. Thus, it is reasonable to conclude that beta cells from different donors secrete different amounts of insulin in response to elevated glucose, and our results obtained with isletOrganDOT cultures prepared from over 60 donors represent natural donor-to-donor variation. Another feature of the isletOrganDOT system is that the cultures retain robust GCIS responses in extended culture. This slide shows data from 2 donors where GCIS assays were performed after 4 or 5 days post-seeding, returned to the incubator for several days, and then the GCIS assays repeated with the same cultures. As you can see, the GCIS responses were well maintained. These data again illustrate large differences in the amount of insulin secreted by cells from these 2 donors. The ability to be able to reuse isletOrganDOT cultures for GCIS assays offers the opportunity to screen cultures for good GCIS responsiveness before testing compounds, and also to batch test compounds using the same cultures. A key aim for the development of the isletOrganDOT system was to establish if the cultures retain responsiveness to GLP-1 receptor agonists, and the next slide shows data from 5 donors where the effects of exendin-4, a more stable analog of GLP-1, on GCIS were investigated. isletOrganDOT cultures from all 5 donors showed enhanced secretion of insulin in response to raised glucose and further elevation of insulin secretion in the presence of 25 nanomolar exendin-4. Note that some donors responded to exendin-4 with just one of the glucose concentrations used, e.g. donor 13, but others showed exendin-4 enhanced insulin secretion in the presence of both 6.7 and 16.7 millimolar glucose, such as donor 12. So isletOrganDOT cultures yielded robust and consistent GCIS responses, and most cultures also responded to exendin-4. The data confirm that the cultures must retain good expression of the glucose transporter, GLUT2, and the GLP-1 receptor. But we wanted to know how expression of these and other key beta cell genes in isletOrganDOT cultures compared with that of native islets. We used QRT-PCR to measure the expression of 7 key genes, and the data are shown on the next slide. The results are expressed as delta CT values. So the smaller the value, the higher the expression of the gene. For native islets from 2 donors, we saw, as expected, very high expression of the proinsulin gene, and lower but still high expression of the gene for proglucagon, a marker for islet alpha cells. Expression of the other genes of interest was much lower but readily detectable. Slightly higher levels of expression of the same group of genes was apparent in isletOrganDOT cultures, indicating that the cells retained expression of these key beta cell genes. Moreover, expression was maintained in extended culture. As you can see here, the first gene expression profiling was done on day 12 of these cells in culture, and then we did further expression profiles on day 21, and the actual expression of the genes changed very little. The same is true of this particular donor on day 11 and then on day 20. We were particularly interested in GPR40 and GPR119 as these are drug targets for new treatments for diabetes. To test if expression of the GPR40 gene translated into functional protein, we investigated the GPR40 agonist TAC875 for its effects on GCIS in isletOrganDOT cultures. This slide shows that the agonist strongly enhanced GCIS. Though TAC875 reached phase 3 clinical trials, its development was eventually stopped due to liver toxicity issues. Nevertheless, GPR40 and GPR119 agonists represent a logical approach to the treatment of diabetes, and our TAC875 GCIS data add weight to the value of isletOrganDOTs for testing novel insulin secretagogues. Whilst current diabetes therapies do provide some level of glycemic control, they often do not prevent the debilitating long-term consequences of the disease. In the ongoing search for better treatments, there is a focus on strategies that aim to preserve the function of remaining beta cells or to replenish beta cell mass. The robust nature of the human isletOrganDOT system means it is well placed to aid research and development in this area. To this end, we aim to develop a human in vitro model to support the identification of new therapies for diabetes, focused on beta cell health and proliferation. The model will be developed using native human islets, as we've established the gold standard for understanding human islet cell biology. We also wish to explore the possibility of replacing native human islets with iPSC-derived beta cells as a means to creating a sustainable and higher throughput screening platform. And finally, we wish to investigate whether the established model, whether it's with native human islets and or iPSC-derived beta cells, can be miniaturized using 3D microvalve printing. To achieve these aims, we are collaborating with Definigens, a Cambridge-based company with expertise in the generation and manipulation of human pluripotent stem cells, and Dr. Jing Yang at the University of Nottingham in the UK, who is an expert in the field of bioprinting. We are also grateful to Innovate UK for the award of a research and development grant to support this work. Our initial studies have focused on testing agents that are reported as stress factors for beta cell health. Published studies report that free fatty acids, in particular palmitic acid, may play a role in the destruction of beta cells in the pancreas of type 2 diabetics. As part of our work, we are investigating the effects of a range of free fatty acids on beta cells in isletOrganDOT culture, using insulin secretion in response to raised glucose as a sensitive endpoint measure of beta cell functionality. To focus on palmitate, the preliminary data shown on this slide confirm that incubation of isletOrganDOT cultures with this fatty acid markedly affects the ability of beta cells to secrete insulin in GCIS assays. We incubated cultures with concentrations of palmitate and then performed GCIS assays after 24 hours, 48 hours or 1 week of treatment. In the absence of palmitate, the cells responded well to glucose at all time points tested. Palmitate had no apparent effect on insulin secretion after treatment for 24 hours, but after 48 hours we observed marked effects on both low and high concentrations of palmitate on insulin secretion from the cells in response to raised glucose. And this was an effect that was maintained up to 7 days. This reduction in GSIS responsiveness doesn't appear to be due to the frank destruction of the aggregated islet cells, as these images show. So these are control cultures after 7 days, and as you can see, the 3D structure of the aggregated islets is readily apparent. But even after incubation for 7 days with high palmitate, we still see these aggregated structures. These encouraging preliminary results confirm published effects on palmitate on beta cells, and the experiments are currently being repeated with islet cells from more donors. So in summary, the human islet organ dot system provides a robust platform for GSIS assays, and the cultures retain functionality for at least 3 weeks in culture. The cultures show consistent responses to glucose, GLP-1, and secretagogues in GSIS assays, and may be pre-screened to ensure experimental suitability prior to committing valuable development compounds for testing. The longevity of the system allows retesting or batch testing of compounds to be done using the same donor. In addition to beta cells, we have demonstrated that alpha and delta cells are present in islet organ dot cultures, raising the possibility for investigation of the biology of these cell types and examination of the effects of compounds on, for example, glucagon and somatostatin secretion. And finally, the culture format may be used with various endpoints, for example, immunocytochemistry, qRT-PCR, and transfection for gene knockdown. That's the end of my presentation. So thank you very much for your attention. I have some questions that have been posted during the course of the webinar. The first question is, do your cultures always respond to glucose and exendin? At least 90% of the islet organ dot cultures that we have set up have responded to glucose, and about 75% of these also responded to exendin 4 in the presence of at least one of the glucose concentrations tested. However, even when we don't see a statistically significant response to exendin compared with the response to the corresponding glucose only, we generally see the same trend. Another question is, can the system explore the biphasic insulin secretory response? Well, yes, in principle it can. What I have to say though is that we optimize the current assay time point to capture the latter sustained secretion of insulin. Another question. Do you have access to islets from diabetics? We work with specialist sites to source high-quality islets for research. So typically these are sites for clinical transplantation and are routinely isolating from non-diseased pancreata. However, we have been able to request isolations from diabetic donors, but obviously these are not as frequent as from non-diseased donors. A further question here. It appears you did not perform DNA normalization of your insulin release data. Why is that? Well, actually we have done that, and we've normalized to protein as well, and we see the same effects. So we do generally believe that it's not the number of cells that differ in the cultures, and it's not the number of beta cells. So the normalization gives us the same overall effect. A further question. After treatment with palmitate, the cultures look more morphologically normal but lose function. Why do they still look normal? The answer to that is that we believe that palmitate is disturbing secretory function and not causing cytotoxicity. In fact, there is evidence in the literature that is likely the case in beta cells. And another question concerning beta cells. How are you planning to stimulate beta cell proliferation in the model? Well, we've already optimized various endpoints in the system, e.g. BRDU staining for measuring proliferation. And now we just need to find the right stimulus for beta cell proliferation. We've explored some GLP-1 agonists, and we will be looking at trying to optimize stimulation with these and other agents such as beta-trophin and possibly harmine. And another question that's come through is how much does our system cost? Well, what we do is we ask our customers to work with our business development directors, who will tailor a study to them, but we can send out approximate figures for how much it would cost to test compounds in the system. The questioners also said, how can they get these islets? I'm not sure if that means the fresh isolated islets or the islet organ-dot cultures. At the moment, we only use the islet organ-dot cultures in-house. We have not tested whether they could be transported to customers' labs and also retain functionality. So that's not something we're doing at the moment. We are open to training customers in how to set up islet organ-dots, and we can point them in the right direction of where to try and source freshly isolated islets. So I have a question here on compound testing in the system. What are the minimum and maximum number of compounds that can be tested with the system with islets from a single donor? Well, the answer to that is really a flexible system. So we have the islet organ-dot service that we offer is set up to provide what we call test plates, and each test plate allows for 6 test conditions. The number of compounds that could be tested depends on the conditions required, such as the number of glucose concentrations to be used and how many concentrations of each compound are to be tested. So if a client wanted compounds to be tested at a single concentration of glucose, then 6 compounds at a single concentration could be tested on each plate. For 2 concentrations of glucose, then 3 compounds could be tested, and so on. As I mentioned, the system offers great flexibility, and we work with our clients in the design of test plate layouts. What I would say is that, as I mentioned in the presentation, is that the organ-dot methodology is proprietary to Asterand, but we would potentially entertain out-licensing or providing a license to clients who want to use it in-house. Another question relating to test compounds. How quickly can you test compounds? Well, we are dependent on suppliers of freshly isolated islets, and although it can be variable, we receive, say, 2 to 3 offers of islets from our suppliers each month. Our usual turnaround time for testing compounds would be 4 to 6 weeks once a project has been commissioned and we have received the customers' test compounds. And there's one more question left. Why do you use these concentrations of glucose? It's fairly straightforward answer to that. The normal blood glucose range is 3.5 to 6.5 millimolar, which would typically rise to about 8 millimolar after a meal. Now, obviously, diabetics who are not well controlled would experience much higher blood glucose levels, particularly after a carbohydrate-rich meal.
Well, thank you, Graham. A lot of questions coming in there at the end. Unfortunately, that is all the time that we have for this webinar today. So for any unanswered or further questions, please do not hesitate to contact us. As previously mentioned, a recording of the webinar will be made available on the Asterand Bioscience website, and a follow-up email will be sent out with a link to this following this presentation. On behalf of Dr. Place and Asterand Bioscience, thank you so much for joining us for this presentation.